some science. Now, we have seen what happens in metaphase 1. Now, obviously, from metaphase, then we go to anaphase. And with anaphase, it's the same thing. The same thing that happens in anaphase of mitosis, it's the same thing that happens here. You separate. And these chromosomes, stroke chromatids, move to opposite poles. Okay? Now, the difference between anaphase 1 of meiosis 1 and anaphase 2 of meiosis 2 is that here, the homologous chromosomes, they will separate. Okay? The homologous chromosomes, they will separate. And each chromosome now is going to be forced to move to the opposite poles. Each chromosome, okay? Not chromatid. We saw that in anaphase of mitosis, they are chromatids that move to opposite poles. But in anaphase of meiosis 1, they are chromosomes that will migrate to opposite poles. Remy, I've seen your question. I'm going to answer. Okay? I'm going to answer. Have no worries. Now the homologs, they are the same homologous chromosomes. Now here, instead of, we saw that in anaphase of mitosis, the centromeres break. The protein molecules which join these chromatids at this point known as centromere, they are the ones which will attach the spindle fibers, so that when this one pulls towards this side and this one pulls towards this side, the protein molecules, the proteins, they are known as kinetochores or kinetochores, which join these chromatids at the centromere, they will separate. Okay? And each chromatid migrates to the opposite pole, to the other pole. But here, the proteins that are responsible for sister chromatids cohesion along the chromatid arms, they allow the homologs to separate. What do we mean by this? You know, you have the homologs, the homologous chromosomes. This is the homologous chromosome. And then there is another homologous chromosome. Now, the homologous chromosomes, we saw that they join at the point known as chiasma, or chiasmata. Now, when they attach on the spindle fibers, maybe this one attaches from here, and this one also attaches from here, and these spindle fibers begin to coil, begin to pull. Okay? They are going to pull these chromosomes such that the proteins which attach to these chromatids, these non-sister chromatids, at the point known as chiasma, they will break down, and the two homologous chromosomes will separate, and they will move to opposite poles, like how you can see here. But so guys, yes. So you can see in metaphase, these chromosomes, they were joined together. They were arranged together on the metaphase plate. But now in anaphase, because this central, now somebody asked how spindle fibers pull. I'm going to talk about it. So when these spindle fibers pull, you're going to cause tension on the protein molecules that joined these two chromatids at the chiasmata. And so when you create tension on them, they will separate. but the centromeres of these chromosomes, they will remain the same. They will remain the way they are. So as a result, these chromosomes will move to opposite poles as chromosomes. Unlike in mitosis, where they will move as chromatids. So guys, yes. Now let me answer somebody's question. Somebody asked that how do these chromosomes, how do these spindle fibers pull? Now these spindle fibers, they are microtubules. Okay? They are microtubules that are made up of fibrous proteins, the actin. Yeah, the actin fibers, probably. But that's too much of you. Okay? That's too much of you. Now, the science is these microtubules, these ones, they will contract. And in the process of contraction, you'll need energy. And the energy, they'll get it from ATP. And you remember in the interphase, where we say that it is the preparation stage. That's where the ATP is going to be produced. The ATP is going to be produced in interphase. And when you have enough, that's when cells will proceed to divide. But when ATP is not enough, then the cells cannot proceed to prophase. And I said, or I asked you that what will this ATP going to be used for? And I remember answering that question when we were studying anaphase of mitosis. But it is the same thing. The ATP is going to be used in contraction of these microtubules. And when these microtubules contract, they begin to shorten. Okay? So the tube which was like this, when it contracts, it shortens. And also this one also shortens. So the more you shorten, the more you create tension, hence pulling these chromosomes, okay, to migrate far apart from each other. Not so, guys. And so that's when you form your two cells. That's when these chromosomes, they will successfully migrate to the opposite poles. And they'll be ready for meiosis 2, for the other division, which is meiosis 2. Any question for me? Please, let's ask. Yeah, it's okay, guys. It's okay, guys. It's okay, guys. So what do I want? Okay. So somebody is confusing between these two. Now, look at this. I might give you such a cell. I draw this and I draw this. Okay. Now, do you really notice the difference? The X is a chromosome. Also here, the X is a chromosome. Now here, it is one chromosome, and the chromosomes have arranged on the spindle equator in a way that when this spindle fiber pulls like this, I think pulls like this, the chromosome is going to divide. The centromere here is going to divide. The same applies to this. The chromosome is going to divide. and each two sister chromatids migrate to opposite poles. The same applies to this. This one is going to pull like this, and this fiber also pulls like this. So the chromosome is going to divide, and each sister chromatid, okay? The sister chromatids, each is going to move to opposite poles. And so that one is metaphase of mitosis. And now, for metaphase 1 of meiosis, do you see how the chromosomes are arranged? Do you really appreciate? Yes, that's good. They are the homologous chromosomes, the homologs. Do you see? It's a bundle. There are two chromosomes, and the chromosomes have arranged themselves that if this one pulls towards this side, and this one pulls towards this side, the chromosomes, they are the ones which will separate, and the two chromosomes will move to the opposite poles. If you can really appreciate that, then you'll be good to go. You will know the difference between metaphase 1 and metaphase 2. You will know the difference between anaphase 1 and anaphase 2. And you will know the difference between prophase 1 and prophase 2 and prophase of mitosis. I hope I've really tried to bring it to the ground level. What's up, guys? Yes. In case of any question, please ask. Now with telophase, it's okay. Now with telophase, this is simple. Now actually, here, it's about cytokinesis and karyokinesis. The cytoplasm divides and the nucleus divides, and boom, you form daughter cells. However, some cells don't undergo telophase. That's a disclaimer. Not a disclaimer, but that's a fact. Some cells don't undergo. You know, with meiosis, there are two successive divisions. And sometimes the divisions, they occur continuously. You undergo meiosis 1, and in the moment after meiosis 1, you proceed and undergo meiosis 2. The moment so you might, after anaphase, the cell might proceed, okay, to meiosis 2. But however, also have other cells that can still undergo telophase 1. Now, in telophase 1, the spindle fibers obviously will disappear. And then the cell divides, is going to divide into two daughter cells. And each of the daughter cells is going to have half the number of chromosomes as compared to the mother cell. And NB, the nuclear membrane might be formed, or the cell membrane might go directly to meiotic division 2 without forming the nuclear membrane. So that's it. So if I give you such a cell, okay, and I've constricted, it will divide, then you know that this is telophase 1. And so I'm going to stop here for today. And in our next lesson, that's when we shall proceed with telophase 1, and then we go to prophase 2, and we finish meiosis, and we see significances. We compare both meiosis and mitosis, and we call it a day. And we call it